Alright, this is the explanation video for the mock test number 2 that you guys took just recently. So question number 1, assume a person is placed on cortisol medication, a glucocorticoid. How would you expect the rates of ACTH and CRF hormone production to change in that person? I would expect both to actually decrease. The reason for this is that cortisol, which is secreted by the adrenal gland, actually has a negative feedback mechanism that affects the pituitary gland as well as the hypothalamus. So the hypothalamus is what is going to release the CRH or CRF. It's corticotropin releasing hormone or corticotropin releasing factor. And then that will stimulate the pituitary to then release the ACTH hormone. Which then stimulates the adrenal gland to release cortisol. So if you have enough cortisol, your body is saying, okay, enough, I don't need any more. Therefore, it will block the pituitary gland and the hypothalamus from secreting those two hormones. All right, carbon particles were injected into the blood of mice. In which of the following places would you most likely find the particles? So this is kind of thinking about in the blood, you've got these particles, and what do you think your body is going to do with them? So most likely you're going to find a lot of them in basically monocytes of the spleen. So this has to do with the reticuloendothelial system, or also named phagocyte system. You have phagocytes in a lot of different tissues of the body that are responsible for removing particles. The monocytes are actually blood phagocytes, basically, and then if they migrate into tissues, they become macrophages. And so this infographic right here is talking about a lot of different macrophages, because these macrophages have already kind of associated themselves with different tissues. So in the blood or in some kind of aqueous solution in the lymphatic system, things like that, you're going to find the monocytes. Monocytes that come in contact with the carbon particles will phagocytose or engulf them and take them to the spleen being a secondary lymphoid organ where the monocytes that turn into macrophages, if they are associated with tissue, can then present carbon particles to other cells, and just it's a way for the body to see if there is actually like a threat, if it were a pathogen or something like that. But in this case, they're just carbon particles. This is also something that will help clean out the body if you've got like those contrast dyes. So if you ever do any kind of like a scan where you need a contrast, you are injected with some kind of dye that helps contrast different tissues or something like that. It will be cleaned out of your blood basically via this system, the reticuloendothelial system. All right, question number 3. Kidneys provide osmoregulation for the body. Consumption of which nutrient creates the greatest need for osmoregulation? So this ends up being protein. One of the reasons is that proteins are really large. So when you have the kidney filtering the blood and actually, creating your concentrated urine, you are filtering a lot of different ions and molecules, salts, nutrients, things like that. You're filtering a bunch of this out of the blood, all right, in the Bowman's capsule and areas like that where you actually have capillaries where the blood can actually kind of cross easily through a more diffusible membrane. So you're getting a lot of these nutrients getting filtered out of the blood. They will go back into the blood if they need to. And as you're moving through the descending loop of Henle, the ascending loop of Henle, and you go into the distal tube, you're still going to get movement of different salts, waters. It really depends on the need for osmoregulation in the blood. And in the end, you're going to be filtering out urea and even more salt and water, and you're really just kind of ending up with like a more concentrated urine. So proteins are really too large to be able to kind of filter across the membranes and into the tubules. And so if you have like a high consumption of protein, this can lead to a buildup of proteins in the blood because they can't really easily get filtered out of the blood and be, basically secreted in the urine. So that will lead to a higher need for osmoregulation. All right, during translation in prokaryotes, the ribosome binds to, it actually binds to the Shine-Dalgarno sequence. This is a little fuzzy. So the Tata box and the enhancer sequence, I have a little picture here that's not that great, sorry. These are actually involved in transcription. So enhancer sequences are going to be different DNA sequences that transcription factors or proteins can bind to, which will help enhance or drive transcription forward. Of course, the Tata box, this is something that will be recognized by RNA polymerase, which will come down and then be able to start transcription if you have other transcription factors there that are required for the initiation. So the Tata box and the enhancer sequences, these are specifically having to do with transcription. The mRNA cap, this is not something that's actually in prokaryotes. It's only in eukaryotes where you have the processing of the RNA after transcription before translation. And then a terminator sequence, this is really actually also kind of has to do with transcription. A terminator sequence is a sequence towards the end of the basically open reading frame of whatever you're trying to transcribe that will signal the RNA polymerase to stop transcribing, to stop making the mRNA. So actually the Tata box, the enhancer sequence, and terminator sequence, these all have to do with RNA transcription. The only things that are really involved in translation are going to be the mRNA cap and then the Shine-Dalgarno sequence, which is actually the answer. This is just a little image showing you. So in prokaryotes, what happens is the small subunit of the ribosome will actually recognize the Shine-Dalgarno sequence and bind to it, and then it'll also associate with the initiator tRNA. So in prokaryotes, it's going to be F-methionine. It will then start scanning down the mRNA transcript until it hits the very first AUG, and then the tRNA will bind much better, and then the large subunit will come in, and you'll get like the full formation of the ribosome, and then translation actually starts. Now in eukaryotes do not have the Shine-Dalgarno sequence. So what happens in eukaryotes is after the mRNA sequence is processed and becomes the mature RNA, it has a 5' cap and a poly A tail. The 5' cap is on the very 5' end. The poly A tail is on the end, like the very 3' end. So what the small subunit of the RNA is actually recognizing in eukaryotes is the 5' cap. And then it'll associate with the initiator tRNA, which is just methionine, not F-methionine. It will then scan down the RNA transcript until it hits the very first AUG. Then the large subunit will come down, and then translation is officially initiated. All right, question number 5. The quaternary structure of a protein is determined by... you have a few different choices on here. So you have to think about the different levels of structure you get from the proteins. So you've got the primary structure that's just going to be really the chain of amino acids. You have the secondary structure, which is going to be different sections of that polypeptide chain, is actually going to... Sorry, my mail just popped in. All right. Different sections of the polypeptide chain are going to start forming structures like alpha helices and beta sheets. All right, so D right here is not the answer because this is actually going to be part of the secondary structure. The tertiary structure, this is where you actually get the 3D folding of the protein. This is when you'll have interactions between different R groups of amino acids that are part of the same polypeptide chain. So A and B are actually going to do more with Um, Probably tertiary structure, could also be secondary structure, because when you start forming the alpha helices and beta pleated sheets, those are associations between close amino acids on the same polypeptide. So that could be kind of either secondary or tertiary. And so really what it's going to end up being is interactions between amino acids of different polypeptide chains. Binding of a coenzyme or cofactor to a protein, this is something that's going to happen after the protein's been folded, typically something that helps the protein, which is going to be acting as an enzyme, bind to its target better and function better. All right, but this doesn't have anything to do with the actual structure of the protein. So the answer is interactions between amino acids of different polypeptide chains. So this is an example of the quaternary structure. So a quaternary structure, you have different 3D folded proteins that come together to form a larger complex. They interact with each other. There are two main kind of structural types that are well known. Collagen, so these will form long fibrous connective tissue, and what you have is 3 entwined protein subunits. So you have these alpha helical proteins, and they just kind of like entwine themselves, kind of like a rope. And then the other example is hemoglobin. This will form a globular shape, and so what you get with hemoglobin is you have 4 subunits, all right, that actually come together to form the quaternary structure. Hemoglobin is, of course, responsible for transporting oxygen as well as carbon dioxide through the blood for respiration purposes. All right, which of the following is not a function of the parasympathetic division of the nervous system? So there's only one of these that is not involved in the parasympathetic division of the nervous system. That is going to be stimulation of the adrenal medulla. So this is a figure in the Campbell Biology textbook, which I think really summarizes this really well. So your choices were constriction of bronchi in the lungs, constriction of the pupils in the eye, slowing of the heart rate. So if you look, you've got constriction of pupils, constriction of bronchi in the lungs, slow heart. All right, these are all part of the parasympathetic nervous system. And then, oops, wrong way. And then simulation of activity of the stomach and the intestines. So within the parasympathetic division, you also have stimulation of activity of the stomach and intestines, stimulation of activity of the pancreas, stimulation of activity of the gallbladder. Everything just starts kind of like moving, in the body, like emptying of the bladder. Yeah, that's fun. And so the only one of those choices that is not parasympathetic, but would actually be the sympathetic division, would be the stimulation of the adrenal medulla. All right, so adrenal medulla, this comes from the sympathetic division of the nervous system. Okay, question 7. You place a growing seedling in an east-facing window. On which side of the plant will auxins accumulate? They will actually accumulate on the west side. So this is why, all right, phototropism is the growth of a plant either towards or away from the light. Typically, if a plant is photosynthetic, which plants are, then they will want to grow towards the light because this enhances photosynthesis within the plant. So if they're going to grow towards the light, typically you're going to have a lot of different hormones that are involved in that kind of behavior, that tropism. And auxins are really big players. Auxins are just chemical molecules, any chemical molecules that promote elongation of stems. And the way the auxins work for phototropism is they tend to accumulate on the darker side and then promote elongation of those cells. So for instance, here is the sun facing this side, so you've got the auxins collecting on the opposite side, like the darker side, away from the sun, and then they will stimulate elongation of the cells on that side, which kind of tends to bend the plant or the stem towards that light source. All right. So if you put the plant in an east-facing window, then the side opposite of that would be the west side, and that would be the quote-unquote darker side of the plant, which is where the auxins would accumulate. All right, which of the following is not a characteristic of all mammalian species? This is placenta. All right, so all mammalian species, we've got a dorsal nerve cord, endotheric, okay, metabolism. We are endotherms. I don't think that's right. All right, we do have lungs and we do have mammary glands as well. So most mammalian species do actually have a placenta, although there are a few exceptions. So the echidnas and the platypus. All right, these are mammals that actually lay eggs. They don't have a placenta like typical mammals. They are the exceptions. All right, suppose the lac repressor of E. coli is mutated so that it never binds to the operator. Which of the following is true? So this goes into how the lac operon is actually controlled in E. coli. You have a couple different main parts. You have the operator, you will have a repressor, you have the promoter region, and you're also going to have lactose itself, of course, which is going to be basically a stimulator of the lac operon. So the lac operon is one that is typically off unless lactose is present or available. And if that happens, then it basically turns the lac operon on. So it's typically off. It is an inducible operon. All right. So if you have a mutation in the lac repressor so that it never binds to the operator, that means the operator is always open. That means the operon is never repressed, and therefore it will always be on and always be making the enzymes needed for lactose digestion. Okay, so lactose digesting enzymes will always be produced. It doesn't digest glucose. That's not what it's for. The lac operon is specifically for breaking down lactose. And let's look at why that is. All right, so this is, again, an inducible promoter. So what typically happens when there's no lactose around is the repressor will be bound to the operator, which means that RNA polymerase cannot actually bind to the promoter and then move to actually induce transcription down the operon. You can't initiate transcription. All right, so it's always off until you have lactose available, and then what happens is the lactose will bind to the repressor. It will actually cause a conformational change in the repressor. Therefore, the operator will be free. RNA polymerase can bind to the promoter and then can move to transcribe all of the enzymes that are part of the operon. All right, so the lactose itself is called an inducer molecule. Now, there is another level to the lac operon. So typically, if you only have lactose present and but you have glucose, all right, glucose is there and lactose, then usually you have a pretty low level of transcription of the lac operon. There's a little bit that is transcribed, but it's kind of leaky, but it's not that great. And that is because the RNA polymerase, while it can bind and actually transcribe the operon, it doesn't really do it very well. So what happens, because this is a way for the cell to be like, okay, I've got lactose, this is like an alternate carbon source for energy, but I've got glucose as well. There's no need to start making a bunch of lactose enzymes because glucose is my preferred source of energy. So I'm going to go for the glucose first. I'm not going to worry about the lactose right now. All right, so again, a little bit of transcription will happen, but it's really not that much. Now, if glucose gets used up and you still have lactose present, all of a sudden the body says, oh, okay, I've lost my primary source of energy. I need to go to an alternate source. Lactose would be that alternate source. So cyclic AMP levels will actually increase, all right, in the E. coli. This will bind to the CAP protein, which is typically inactive unless it is associated with cyclic AMP. This complex will then bind to the promoter. So what it does is it will actually stabilize RNA polymerase on the promoter region, and it will kind of drive it to go through transcription. So you'll get a lot more of the mRNA synthesized from the lac operon when you have low levels of glucose, and therefore this cyclic AMP cap complex is associated with the promoter and helping stabilize RNA polymerase. So that's like the second level of lac operon control. All right, you examine a leaf cross-section under a microscope and find many loosely packed cells. These cells have many chloroplasts and relatively thin cell walls. These cells are what? The thin cell wall is a pretty good indicator of what we're talking about. These are parenchyma cells. All right, so parenchyma, they have relatively thin primary cell walls. They lack secondary cell walls. They have lots of chloroplasts in the leaves, and they also will make up amyloplasts in the roots and stems. So they have specialized functions in different parts of the plant. Then you have the collenchyma cells. So you got collenchyma. These guys are grouped in strands. They, once elongated, they have thicker primary cell walls, and they lend themselves to flexible support for the plant. And then finally, you've got the sclerenchyma, all right? So these guys have really thick secondary cell walls. They contain lots of lignin, and these help support plant structure, all right? So these guys, the collenchyma and the sclerenchyma are more about structure. The parenchyma have more of these specialized functions. For instance, in the leaf, all right, you guys have probably heard of the mesophyll cells in the leaf. They are responsible for a lot of photosynthesis that goes on in the leaf structures. Mesophyll cells are a type of parenchyma tissue, type of parenchyma cell. All right, so you got the palisade, mesophyll, or parenchyma, and then you have the spongy parenchyma or mesophyll. So that is the correct answer because, of course, what we're talking about are the mesophyll cells that are in the leaf. All right, for the biomass pyramid shown below, choose the correct statement. All right, so typically if you see a biomass pyramid, you would see the primary producers actually having a much larger biomass than the primary consumers, and then you'd have even a smaller biomass for the secondary and then smaller for the tertiary. So it kind of would look like a pyramid, like with the thicker base and then the thinner top. But this one is actually opposite, all right? So B can't be right because biomass does not decrease with higher trophic levels. When you go from primary to producer to primary consumer, you're going higher in the trophic levels, and obviously the biomass is increasing because the size of the box is representing the biomass. Humans represent the lowest trophic levels. That's not true. We are not producers. So producers are organisms that are photosynthetic. They can produce their own food, their own sugars. Humans are consumers. We have to actually consume other organisms to get nutrients, to get sugars, to get food. All right, so we're not producers. This biomass pyramid is not possible. That's not actually true. It is possible. So the answer is actually producers reproduce quickly, but are also consumed at a rapid rate. So typically, this is going to be the type of pyramid that you would see. This is where what you see in most ecosystems. Primary producers having the largest biomass, and then primary consumers having less, and then secondary consumers even less, and tertiary even less. So less and less and less as you go up. But some aquatic ecosystems can actually have this opposite type of structure, like the upside down pyramid. So for this, the primary producers would represent phytoplankton, which are basically algae, photosynthetic algae that float in the water. And then the primary consumers represent zooplankton, like tiny, tiny, tiny little animals, little microscopic organisms. So what is really interesting about this level, or the way it's set up, is in these particular types of aquatic ecosystems, what happens is while when you add up the biomass of the phytoplankton, it is less than the zooplankton. However, it can work because the phytoplankton can reproduce so quickly. And they're consumed at a rapid rate, but they can reproduce so quickly they can they can keep up with the rate of consumption. All right. Okay, which of the following would be most likely to increase competition among the members of the vole population in a given area? All right, so what will increase competition among the members of the vole population? All right, an increase in number of hawk predators, that would just decrease the population. All right, because those are predators, they are going to go after different voles. That's not really going to increase competition. If you decrease the size of the population, you actually decrease competition because that leaves more resources available. An increase in the reproduction rate of the voles, that is definitely something that could actually increase competition. Increase in the reproduction rate would lead to an increase in the population size. Increase in population size without actually increasing the resources available would lead to more competition. Increase in temperature, that is kind of variable depending on what you're really looking at. An increase in food supply. This should not increase competition. If anything, it would decrease competition because you're not increasing population at this point, but you're increasing your food supply. You're increasing your resources. So you would have less competition because there'd be more food available. An epidemic of rabies within the vole population. Again, this is something like A, that would decrease the population size of the voles, which would then decrease competition because lower population, while the resources stay the same, would mean less animals trying to use the resources that you're trying to use, so less competition. So it is an increase in the reproduction rate of voles. This is the only one that would actually increase the population size while the resources stay the same. Here's a little vole right here, this little guy. All right, so when we talk about competition among members of the vole population, we're talking about intra-specific competition. This is competition within organisms of the same species. Interspecific competition, this is going to be organisms from like two different species that are actually competing with each other. So when you have intraspecific competition, typically you'll have this kind of like wavy pattern, like you'll have increased population over time and then decrease, increase and decrease. What's happening is population increases, so then you have more competition, then something happens, the population decreases, and you have less competition, all right? So you have this continuous cycle, and that's why the population size changes, that's why the competition can change. It's not always going to be like just constant. There's always going to be some kind of cyclical pattern going on. Interspecific competition, this is just showing you two different studies that were done or experiments showing population size of two different species. When they're grown separately, they both reach pretty similar population sizes by day 10. However, when they're grown together, you have one that reaches a normal population size, the other one kind of goes down and almost dies off. And this happens because typically when you have competition between different species, there usually is going to be one that can outcompete the other. And if this guy can outcompete, and then this guy can't really keep up, and then it'll suffer. It'll suffer in the process. All right, tobacco mosaic virus has RNA rather than DNA as a genetic material. If the RNA genome from a tobacco mosaic virus is mixed with proteins from a human rhinovirus, the result is a mixed virus. If that virus could infect a cell and reproduce, what would you expect the resulting viruses to be like? So really, tobacco mosaic viruses. Yeah, because what you did is you really you took the RNA genome, all right, mixed with proteins from the human rhinovirus. So you took proteins from a different virus, you mixed it with the RNA genome of the tobacco mosaic virus. But the genome is what is like coding for making new viruses. All right, so adding proteins, that's not really going to do anything. Retroviruses, these guys have RNA genomes. They also contain reverse transcriptase. And so basically what they can do is they can take the RNA and basically reverse transcribe it into DNA. And then the DNA itself is then transcribed, translated, and you get assembly of the new viral particles. This is just a little infographic showing viruses will actually, sometimes they can fuse with the membranes, and sometimes they can like inject their material. But the main purpose is to get the genetic material into the target cell, releasing the genetic material, maybe making that reverse transcriptase protein, getting the cDNA to then maybe integrate into the host DNA genome. And then basically the host will run through transcription translation and basically make new viral particles for you. So the viral genome is really what's encoding like how the new virus particles are made. Therefore, you would be getting back to tobacco mosaic virus. All right, question 14. An amphipathic molecule is a molecule that has, this should be pretty easy and straightforward for you guys. Amphipathic molecules have a hydrophilic and hydrophobic part. All right, so a really famous example is a phospholipid. All right, so the phospholipids that make up all your cell membranes, they have a hydrophilic head. This is where the phosphate group is, and also the glycerol backbone. And then they've got the two fatty acid chains coming off, which make up the hydrophobic end. All right, so they have hydrophilic and hydrophobic properties, and that is the definition of the amphipathic molecule. All right, the function of a molecular chaperone is to do what? So molecular chaperones really, excuse me, molecular chaperones are really part of proper protein folding, and a lot of them actually will function in trying to rescue misfolded proteins. So if they find misfolded proteins, they will unfold them. At first, they try to save them instead of just, tagging them to get chewed up. They'll try to save them. They'll unfold them back to the primary structure, and then they'll try to refold them into the proper shape. All right. Let's see, what else do we have? Act as an energy source during polymerization of amino acids into a polypeptide. No, definitely not. Because a molecular chaperone is not an energy source. Act as a carrier molecule and bring activated monomers to a polymer. No, it doesn't do that either. It's not like a carrier molecule for trying to put polymers together. Bind to specific structures on the polypeptide in order to assist the folding of a protein into its correct three-dimensional shape. So this is kind of partial. So it does assist in the folding of a protein into its correct three-dimensional shape, but it could be the part that says binding to specific structures on the polypeptide that may be wrong, because typically these chaperones can kind of work in a more general fashion. They're not necessarily going to target certain proteins. They definitely do not act in transporting rRNA from the nucleus to the cytoplasm. That is not their job. So this is looking at, you've got some unfolded proteins. So you have, there are some different types of chaperones, and they will function in different parts of protein folding. So for instance, when you are first going through translation, you will have some chaperones that actually will assist in the folding of the proteins that are newly emerging from the ribosome. So these are ribosome-associated chaperones. You get the native folded proteins. These guys, maybe something happens, they get misfolded, unfolded, something happens. You have different chaperones. Another name is heat shock proteins. So these are studied a lot in yeast. And what they do is they will try to help refold the proteins back into their native form. So sometimes when you have misfolded proteins, you can get aggregation. So the misfolds will actually start clumping together to form aggregates. You have certain heat shock proteins or certain chaperones that will actually help protect from aggregation. And then you will have certain ones that will help with trying to disassemble the aggregates and kind of get them back into individual misfolded proteins to then get back into the native proteins. If worse comes to worse, then the proteins are tagged and then they get chewed up by the body. All right, ethidium bromide is used in electrophoresis of DNA fragments because it makes the fragments visible under UV light. This is a pretty basic concept for, typical biology if you're doing biotechnology, things like that. It doesn't really do anything with mobility of DNA in the gel. That all depends on the mass of the DNA fragment. It does not increase conductivity of electricity through the gel. No, it does not do that. You have electrodes for that. It does not do anything with the size of the fragments, and yeah, it doesn't maintain a stable pH either. So really what ethidium bromide does, it is called an intercalator. So this is ethidium bromide right here, and it's an intercalator because it will actually insert itself in between your base pairs. So here's a DNA molecule right here. These lines represent the hydrogen bond connection between base pairs. So this might be like a G, here's a C, here's an A, here's a T. So ethidium bromide can actually insert itself in between the hydrogen bonding that you have between different base pairs. And when you concentrate it like that and then you expose it to UV light, it will fluoresce orange. So it's really easy to see where your DNA is. It's kind of like what a gel looks like when you do that. This is just like the overall technique of electrophoresis. So you have the power source, you have this agarose gel matrix material, kind of like Jell-O, but a little bit firmer. You will add your different samples to the wells. You turn it on. These samples, DNA is negatively charged, so it will always migrate towards the positive electrode. All right, so you always have to remember that. You have to be really careful if you do gel electrophoresis. If you hook the electrodes up opposite, the DNA will migrate out of the cell in the opposite direction and basically get lost in the buffer that you have the gel kind of floating in. So they'll migrate. You typically have some kind of blue dye so you can see it with your eye, where at least kind of like there will be like just like a little blue line, like the dye will migrate through the gel as well, but you can't see the individual bands yet. So what you have to do is actually put the DNA gel under a UV lamp, and then the ethidium bromide will light up orange, and you can see the individual bands. Ethidium bromide is also something you have to be really careful with. It is a mutagen. So because it's an intercalator, which means it can insert itself in between different base pairs, if your body starts going through replication, it may mistake this ethidium bromide molecule for like maybe a base pair collection or a connection, and excuse me, may end up like adding a base there or something like that. So it may end up causing mutations to the DNA. So if you ever work with this, wear gloves. Do not ever touch it with your bare hands. All right. The species Homo sapiens first appeared in which geologic epoch? All right. So we've got a few different ones. It's the Pleistocene. All right. So here is an infographic kind of looking at the different time periods. We have Miocene here, we have Pliocene, and we have the Pleistocene. Hopefully I'm pronouncing that correctly. Homo sapiens, we are Homo sapiens, and this is like the basically most recent, oh gosh, what am I trying to say? Most recent, I'm trying to say, like, I don't want to say outcrop, but like most recent, I don't know, cousin of the bipedal organisms. Like basically it's us. They're modern humans. Guess what I'm trying to say. This is kind of like a little timeline of different types of ancestors that are also kind of related, connected to Homo sapiens. You have the Homo Neanderthals, basically. You have other types of Homo species. And then you have other types of genuses and species. All right, the Homo sapiens, so we are the most short-lived, like we are the most recent, I guess, emergence of the human race, and we are the only ones that are really left. Although, just FYI, everybody has a percentage of Neanderthal DNA within their genome, because when Homo sapiens first appeared, they were actually mating and intermixing with the Neanderthals, which I think is pretty cool. So we are the most recent, we are the shortest lived so far, and so we are in the most recent kind of time period right here. All right, which of the following marine communities would be least affected by volcanic eruption or a meteor impact that injected enough debris into the atmosphere to reduce sunlight by 50% for 2 years? All right, so 2 things you want to think about with this. You're reducing sunlight, okay, so if you have photosynthetic organisms that are part of that marine community, then that would not be a good thing. But also, if you reduce sunlight, which means you reduce photosynthesis, that means you reduce oxygen. Okay, you reduce oxygen output. So you also have to think about the organisms that actually use oxygen. All right, and it's like for 2 years. This is a very long time. So it is actually a community that doesn't really have an oxygen-rich environment and is filled with organisms that use something other than sunlight for energy production. All right, so this is going to be the deep sea vent community. So here are the other ones that are kind of listed. You have the estuaries. These are basically the transition areas between the river and the sea. So it's kind of a mixture of salt and fresh water. You have a lot of different organisms, but notice that you have grasses and algae. These are photosynthetic organisms, so they need sunlight. Then you have worms, oysters, crabs, and fish. All right, so these are going to be organisms that need oxygen. The pelagic zone, this is just open blue water. All right, so you have a couple different zones. So you got a photic zone and an aphotic zone, which means the photic zone is where light hits. The aphotic zone is where no light hits. So within the pelagic zone, you do have lots of organisms that don't really rely on sunlight. However, you do have other parts of the pelagic zone that you have organisms that do rely on sunlight. Phytoplankton, this is a photosynthetic algae, cyanobacteria, same thing. And then a lot of other types of organisms. Coral reef, this is like the most diverse type of aquatic ecosystem. Tons of populations of marine organisms all living around these coral communities. They consist of lots of different types of algae, again, photosynthetic organisms. You have corals, which themselves are actually animals. And then you have fish, invertebrates, and just a ton of different organisms living around those communities. Now, benthic means things that are along the sea floor. All right, so all along the sea floor, you're going to have a really big mixture of organisms. You will have different seaweeds and algae. You'll also have lots of different invertebrates, and you can even have some fish and things like that. So again, photosynthetic organisms. The only one that really does not have lots of oxygen available and really doesn't rely on any kind of photosynthesis is going to be the deep sea vent. These are deep sea hydrothermal vents in mid-ocean ridges. Super, super, super deep. Really, really, really hot. All right. So these deep sea hydrothermal vents are super hot. And you have a lot of different types of organisms that are actually chemoautotrophs. So they are autotrophs. They can make their own food, but chemo. So the source are going to be different chemicals that are spewing from these hydrothermal vents, like sulfur, hydrogen sulfide, things like that. So this is really the only one that would not really be bothered by a reduction of sunlight or a reduction of oxygen. All right, all of the following are modified stems except, so this one, they are all kind of modified stems. So this doesn't really have a correct answer. These are pictures of a lot of the different ones that are discussed. So you have the stolon. This is a shoot that grows along the ground and produces roots at its nodes. Let's see, which right here. All right, so here's like here's a stolon right here. You got the tendril. This is a thin, spirally coiling stem that attaches a plant to its support. So typically, if you have like those creeping vines or things like that, you'll have like a wooden dowel or something like that, and they can kind of wrap around and start crawling up. You have the rhizome. All right, so a horizontal underground stem of some plants that sends out roots and shoots from its node. An example of this is actually ginger. So if you guys get fresh ginger and, I don't know, forget about it, and after a while you notice that there's like a little shoot coming out of it, That is a rhizome. So it's got little nodes where you can actually get roots or shoots coming from. You've got the tuber. All right, so tubers are fleshy, thickened underground stem of a plant. These usually contain lots of starch, so your potatoes, things like that, are tubers, and also arrowroot. And then finally, and from these, all right, so these are underground stems. From these, you can get lots of roots coming out. Like if you place it in water, like you'll have a bunch of roots coming out of it. And then finally, you've got the bulb. The bulb-shaped root portion of a plant, such as a tulip, from which the rest of the plant can be regrown. All right, so here's the bulb right here. So you've got roots that can come out from this side, and then you've got, the upper part that can come out from this side. So really all are examples of modified stems. All right, 20, which statement describing plant structure is true? The sclerenchyma cells provide support for the plant body. I kind of covered this in a previous question. So let's look at some of the other ones. So for instance, stomates or stomata are pores in the roots that allow nutrients to enter. No, the stomata are pores, but in the leaves. So the point of the stomata are to allow the exchange of gases like CO2 and oxygen, and then also transpiration of water. Let's see, which one? Wood is formed from old epidermal cells. Wood is actually formed from xylem. All right, so when you have like the trunk of a tree, the inner portions, the wood, are the xylem. Then you've got the vascular cambium, and then the outer bark portions are actually phloem. All right, so that is not epidermal cells. Parenchyma cells have thicker cell walls than other plant cells. Remember when we talked about the parenchyma, the sclerenchyma were the ones that have the thick cell walls. The parenchyma have really thin cell walls. And they lack the secondary cell walls. They have very thin primary cell walls, and typically have specialized functions. For instance, they have lots of chloroplasts in the leaves for photosynthesis, or they contain lots of amyloplasts in roots and stems for storage of starch. Let's see. Yeah, so typically the sclerenchyma, these are going to have thicker cell walls. They contain lignin, all right, which is like a pretty kind of structural protein, and this helps support plant structure. All right, so that is your answer. All right, the Hardy-Weinberg equilibrium principle. If this assumes all of the following statements except what? All right. The whole point of Hardy-Weinberg equilibrium is that everything is in equilibrium, so therefore remains stable. All right, so A is fine. The frequency of dominant recessive alleles for a given trait in a gene pool of a large population can be determined mathematically. That's true. There is a Hardy-Weinberg equation, so you can actually determine that mathematically, especially in a large population. That's one of the requirements for Hardy-Weinberg equilibrium. Genotype frequencies in large population are unaffected by Mendelian segregation and recombination of alleles. So basically it's saying that you're not going to get like a shift in different phenotypes and genotypes. They are going to remain stable. For a given trait, the frequency of the genotype of the heterozygous population can be determined mathematically. Again, yes, there is a Hardy-Weinberg equilibrium equation. That leaves us with, for a given trait, the frequency of dominant alleles is greater than that of recessive alleles. Hardy-Weinberg does not dictate the frequency of dominant versus recessive. That is not part of it. What it's really talking about is everything remaining stable over time. All right, so dominant versus recessive, that is really dependent on like what the beginning genotype was, and then that'll remain stable over time. Of course, you have to have some different requirements for Hardy-Weinberg equilibrium to happen. You have to have random mating, all right, so no like sexual selection, anything like that. No migration, no genetic drift, no mutation, and no natural selection. And of course, you also need to have those large populations. And then here is the equation. So mathematically, yes, you can determine frequencies. All right. Professor Jones is running an experiment with his graduate students in his physiology lab. One group of students drinks a liter of water, another group drinks a liter of coffee, and a third group drinks a liter of concentrated salt solution. The volume of urine produced by all individuals in the 3 groups is measured over a period of several hours. At the end of the monitoring period, which group will have produced the greatest volume and which one will produce the least? So coffee drinkers will produce the most, and salt solution drinkers, which sounds just really gross, will produce the least. All right, so salt solution, you guys probably kind of picked up on that, because if you have super high salt, then you're going to get retention of water, because your body's going to try to stabilize the osmolarity of the blood and of the body and stuff, and so it'll try to retain water. Let's see. Yeah, so people who did the salt solution will not produce the most, so you can automatically knock out C and D, and there's no difference? Yeah, right. So you can basically knock out C, D, and E, and it takes it down to A and B. So it's just between, and basically those both say the drinking coffee will produce the most urine. The only difference is the least is either coming from water or the salt solution. So that should have been pretty straightforward for you guys. So this B. So why is caffeine, or why is coffee something that would make you produce more urine? Caffeine is actually acts as diuretic. It increases urine output, all right? And so here's a few different things that caffeine will actually do. It increases blood vessel vasoconstriction, so it increases your blood pressure, increases acid secretion, changes metabolism, where it increases lipolysis, decreases glucose uptake, increases hepatic glucose production. It can increase wakefulness with the adenosine receptor antagonism, and it can increase your shortness of breath, but also it can increase your urine output. All right, so that is probably going to make you have more urine output than just drinking water itself. Now with the salt solution, this kind of goes into this osmoregulation pathway. So you will have blood osmolarity increasing, like such as if you sweat profusely or if you gulp down a salt solution. Osmoreceptors in the hypothalamus will trigger the release of ADH from the posterior pituitary gland. The ADH will then actually stimulate the collecting ducts to actually release water to get reabsorbed into the blood. So you're basically retaining fluids. So here's the collecting duct right here. And then the other thing that will stimulate will generate thirst. So it'll make you want to drink water to reduce your blood osmolarity, and then hopefully you'll return back to normal. Okay, a protein is usually tagged for degradation by proteasome activity by which of the following proteins? So it is ubiquitin, all right? So caspase, this is a protease that's actually involved in apoptosis. There are a lot of different caspases in the cells, but they are involved in apoptosis, which is programmed cell death. So they are not really specifically looking to tag proteins for proteasome, which the proteasome is what it's doing is it's chewing up proteins. Kinases, these are enzymes that are transferring a phosphate group. So kinases will add a phosphate group to a protein, either to activate it or inactivate it. Protease, this is an enzyme that will actually hydrolyze proteins. It will clip the proteins up and break those covalent bonds between amino acids. Ubiquinone, all right, so this is actually part of the electron transport chain. So another name is coenzyme Q10. And this is a carrier, an electron carrier, part of the electron transport chain. So you're finally left with ubiquitin, all right? So this is called ubiquitination. What happens is you've got, here's your substrate right here, all right? So you add these ubiquitin molecules to your substrate. You actually add like a string of ubiquitin molecules. And what this does is this will tag that substrate, that protein, to be basically taken to the proteasome, and then the proteasome is this cool, like cylindrical structure where it has like this cap to it. The cap opens and the tagged protein goes in, and it's like a shredder. It's basically acting like a shredder. It'll chew up the protein into tiny pieces, and hopefully those get recycled back to making like other proteins. All right, which of the following statements is true of territorial behavior? So you've got the intruders and the owners, all right, in this scenario. So it says contests are seldom ritualized. Yeah, right. I'm sure you guys have seen a lot of examples of different types of organisms defending their territory. A lot of times they'll have some kind of ritual. They'll display themselves. They'll do something, and then they'll approach each other and all that stuff. So usually it is pretty ritualized. The other thing is what the answer is owners defend mainly against conspecifics. Because the owners are familiar with the territory, so they are the ones that are typically going to not only win the majority of confrontations, although that also kind of depends on really the size and the fighting ability of the intruder and the owner. So just because an intruder doesn't mean it's always going to win, or just because an owner doesn't mean it's always going to win. Really depends on like the size and the ability of the intruder and the owner. But owners are going to typically escalate the battle because they're the ones who were like, this is my spot. I have put a bunch of energy into it. I'm not letting it go, kind of thing. Yeah, so owners are going to be more familiar with the territories, and territories are going to be like these fixed locations. And so they will be more likely to escalate the battle. An individual that has gained a territory is often difficult to dislodge. So once they've gained the territory, typically it's much harder to dislodge them. And a territory is definitely worth more to the owner than the intruder because the owner is already familiar with it. So it's definitely like the owner has already put work and energy into like becoming familiar, doing things with resources like maybe making shelters or, making some kind of display for females or something like that. This is a cost or benefit chart against territory size. There's kind of like a trade-off, all right? So the larger your territory gets, then the higher the cost is to actually defend it, and it becomes not really beneficial. There's like a sweet spot, like there's an optimum level where you're getting a benefit, you're gaining a benefit, but at a certain point that kind of levels off, and the cost of defense greatly surpasses the benefit that you gain from like having that territory. If the territory is too big, it's hard to defend. You could get like a bunch of different intruders coming at the same time. So you got like a little sweet spot. And that will change depending on the size of the organism and things like that. When it says conspecific, this means members of the same species. All right, so mainly when you've got territorial behavior, the owners are going to be defending against members of the same species, competition. All right, you extract RNA from liver cells and then carry out an agarose gel electrophoresis of the liver RNA. The RNA fragments are then transferred to an RNA binding membrane, nitrocellulose or nylon, using capillary action. Next, you hybridize a probe for gene X to the RNA on the membrane. Which of the following statements regarding your experiment is true? So, you are trying to determine how many copies of gene X are in the liver cells. You don't really know because you're looking at RNA. You're not looking at genomic DNA. All right, so if you're looking at how many copies of the gene are present, you need to look at DNA. You're trying to determine if the gene X protein is present in the liver. Again, you're looking at RNA, not protein. So the only way you could tell if the protein is present is to actually look for the protein itself. You are trying to determine if gene X is expressed in the liver. Yes. So what this means is when a gene is expressed, that means it is actively being transcribed. And if it's actively being transcribed, it's likely being translated. But if you're looking at expression levels, you can really just look at the RNA levels because that'll show you how high transcription is going because of the amount of RNA that's accumulating. You are trying to determine the chromosomal location of gene X. Again, you're not looking at DNA, so that RNA wouldn't really work for that. And then you are trying to determine whether gene X has a mutant sequence. Again, that is not really going to help with just hybridizing a probe to it. You would really have to sequence the DNA to see if there's like a mutation somewhere. All right, so here, everything I just kind of talked about, basically. This is kind of what's going on with northern blot. So this is the type of experiment that was done with RNA that was discussed in the question. You have a sample, you collect total RNA from the sample, you basically run it on an agarose gel. The RNA is separated by size of the bands, and you transfer northern blotting the RNA to a membrane. Then you take the membrane, you wash it with these probes that are specific for the gene of interest you're looking for. And typically they're labeled, you might label them with fluorescence, you might label them with like a radioactive molecule, something like that. You wash away what has not been bound to the membrane, and then you will visualize. If you're visualizing on an X-ray film, that means you were using radioactive probes. So that is a northern blot. Southern blot is basically the same thing, except instead of running RNA, you're running genomic DNA. All right, so that's southern blot. You can still probe with labeled probes for your gene of interest. The northern blot will tell you if the RNA is being made for your specific gene. The southern blot will tell you if the DNA is present in the genome. Does not tell you anything about whether it's being transcribed or translated at the time. If you're looking for protein, you're going to do a Western blot. So Western blot is an analysis of protein, and for that, you're actually using antibodies, not a labeled probe. All right, so you're going to actually use antibodies specific for the protein, and then they will have some kind of fluorescent tag, and then you can use a fluorescent microscope to see what is there. All right, a toxin exists that inhibits the enzyme succinate dehydrogenase. If this were used in an experiment to stop respiration, how many net NAD plus and ATP molecules would be generated from each molecule, or from each glucose molecule, up to that stage? All right, so the answer is 6 NADH plus and 4 ATP. So let's talk about why that is. In glycolysis, all right, for every glucose molecule, you get 2 ATP, okay? You get 2 pyruvate molecules coming off of glycolysis. Once you go through the process of pyruvate oxidation, you're going to get 2 NADH, so 1 NADH per pyruvate molecule. So already, all right, oh, and sorry, forgot to mention, in glycolysis, you also get 2 NADH plus. Sorry, forgot to mention that. So in glycolysis, you've got, and through pyruvate oxidation, you've already got 4 NADH plus molecules, and you've got 2 ATP molecules. This is before you even enter the citric acid cycle, all right? Now, here's the citric acid cycle. So remember, you have a toxin that inhibits succinate dehydrogenase. That is right here, okay? So basically, if you have a toxin that inhibits this, it'll stop the cycle right here. So let's see what we can get up to that point. So you've got the formation of citrate. Here's the acetyl-CoA coming in from the pyruvate oxidation. Here's the citrate to isocitrate. And then as you're moving through, you get 1 NADH molecule, 2 NADH molecules, but that's only for 1 acetyl-CoA. And remember, you get 2 pyruvate molecules from glycolysis. Therefore, you will get 2 acetyl-CoA molecules coming from the pyruvate oxidation, coming from 1 glucose molecule. So you get NADH, you get NADH, and then you basically have this part right here where you have a GDP typically transforming into GTP, but then the GTP can be used to transform ADP into ATP. So you can get another ATP molecule right here, right? So basically what you end up with is 6 NADH plus molecules. That's weird. And you would get 4 ATP molecules, but that actually, I don't think that's right, because with 1 acetyl-CoA, you would get NADH, NADH. So you get 2 more NADHs and then 1 ATP with 1 acetyl-CoA molecule. So that would make 6 and 3. And then if you have the other acetyl-CoA molecule coming through to like complete what you get from 1 glucose molecule, you'd have another NADH, another NADH, and another ATP. So that would bring you up to 4 ATP. But that would bring you to 8. Interesting. 8 NADH molecules. Oh, goodness. So, honestly, that should probably be 8 and 4, not 6 and 4. I would say that's 8 and 4. Well, close enough, I guess. All right, question 27. Which of the following characteristics is an adaptation of temperate climatic zone plants? All right, so you have a couple different adaptations I'm going to talk about. Remember the temperate climatic zone. You've got temperate grasslands and you have temperate broadleaf forests. So focusing on really the broadleaf forests, they can get a pretty good amount of rainfall every year, and the temperatures tend to be warm. However, the thing with the temperate forests is they'll have hot and humid summers, but their winters will have typically lots of snow, so still a lot of precipitation, and we'll get down to, like, on average 0 degrees Celsius, so like kind of right at that freezing level. So dormant buds, this is going to be the characteristic adaptation for this zone of plants. That's because when the plants are exposed to this cold winter weather, they will go dormant so they can like kind of stay alive, but they're not really actively growing. And then when the temperature conditions kind of get to better levels, warmer temperatures, more humid conditions, then the buds can now start growing into like new stems and things like that. So here are some of the other adaptations they have listed. They got these drip tips. So the drip tips are typically found in rainforests that have like a lot of precipitation. This is a way to allow water to drip off the leaf without causing a lot of damage or breakage. If the water kind of sits on the leaf for a long time, it could weaken the leaf and cause damage, or if you get like a lot of water pooling on the leaf. You also got sunken stomata and succulent leaves. These are kind of both found in similar types of climates. These are going to be found in more arid climates, all right? So the sunken stomata, this is how it looks right here. So here is like typically the stomata would be like right here on the surface, but in certain types of plants, it's actually sunken down. So here's going to be the guard cells right here, and you've got this little depression right here. So here's the surface, but then here's where the actual stomata is, or the stoma. And what happens is you've got this little depression, and this actually helps protect the stomata from high winds. So when you do have water evaporation, you don't have wind like pulling the water out faster. And also when you have like water molecules kind of, pool right here, it helps them stick around a little bit longer in case they need to get reabsorbed. With succulent leaves, these remain thick, fleshy, and engorged. So typically if you like cut a succulent leaf, it'll have like a bunch of, like if you think of like an aloe plant, a bunch of like, like a sticky stuff in it. And it helps the plant retain water in arid conditions. So succulents do really good in like hot, low moisture, arid conditions. And finally, very shallow roots. Shallow root systems are typically found in northern coniferous forest biomes. So these are going to be the biomes that like have kind of like the lower temperatures. This can be due to the soil being heavily leached, and also if you have permafrost underneath the top layer. So permafrost is going to be like frozen ground. The roots can't really get through it, and so they tend to be more shallow. Okay, which of the following statements is true? So this has to do with short day and long day plants. And this is actually pretty straightforward if you kind of just know the background of short day and long day plants. A long day plant will flower if it has over a half day of darkness interrupted by a short flash of light. So here's the difference. There is a critical night length. All right, so short day plants, which are also typically long night plants, quote unquote, because long night plants is like mentioned in one of the answers. So short day plants, they flower when a period of darkness exceeds the critical night length. All right. So it'll only flower if it has a long enough night. So if it does not exceed the length, or if you have that short flash of light, it will not flower. All right. So you have short day plant will flower if it has over a half day of darkness interrupted by a short flash of light. No, it will not flower because you don't get that long enough night length. Long night plant, it's basically saying the same thing because the short day is the long night plant. And then a long day plant will flower if it has over half a day of uninterrupted darkness. So the long day plants, they flower when the period of darkness is less than the critical night length threshold. So if it's less, or if you've got that little flash of light, you will get flowering. However, if that darkness goes above that halfway point, then you don't get flowering of the long day plants. All right, which of the following is not a second messenger? So these are all pretty straightforward second messengers except magnesium ion. So calcium ion, diacylglycerol or DAG, inositol triphosphate, IP3, or cyclic AMP. Here are some different pathways that these second messengers are involved in. So cyclic AMP can work with G protein coupled receptors. This is a very kind of common, famous second messenger. And remember, these second messengers are small, like non-protein molecules that can amplify the signal of some kind of stimulus. So you got the first messenger, which is like the actual stimulating molecule, the signaling molecule. Second messenger is going to be something involved in that response that will help amplify the signal. It's typically really small, it can diffuse through the cell really quickly, and it can really help amplify the signal quickly. Here is showing you the other three secondary messengers, second messengers. You've got another G protein coupled receptor. This time you've got phospholipase C, which is the enzyme that is activated. What happens is you have PIP2, which is basically a phospholipid, has a little cap on it right here. This phospholipase C will actually clip it, all right? So what's left afterwards is DAG, all right, the diacylglycerol, and IP3. So DAG itself can actually act as a secondary messenger in other pathways. IP3 will diffuse through the cell and will actually come in contact with these ligand-gated calcium channels. So once IP3, it's an IP3 specific gated calcium channel really. Once the IP3 binds, the gate will open, calcium will flood into the cell because it's higher concentrations in the endoplasmic reticulum than it is in the cytoplasm of the cell. It'll flood into the cell, and then that will cause cellular responses based on the original signal. So no magnesium ion. All right, the reduction phase of the Calvin cycle is equivalent to which of the following processes in reverse? So remember what the calcium cycle is doing, and the reduction phase specifically is reducing something. So think about something that is then oxidizing, not reducing. That would be the opposite, so reduction, oxidation. So the answer is actually the Krebs cycle. All right, so here's the Calvin cycle, here's the Krebs cycle. They look pretty similar, at least because they're, you know, these cyclical pathways where you get recycling of molecules. In the Calvin cycle, you have reduction of CO2 to glucose. So you start with CO2. You have carbon fixation, and then you've got reduction, a series of steps where NADPH is reduced or is oxidized to NADP, and so you are actually reducing your carbon molecule, and then you end up with one molecule of the G3P coming off to go on to help form glucose. The rest are recycled and go back into the whole cycle. So you're going from ATP to ADP, so you are taking energy to do it, and you're going from NADPH to NAD+. So you're oxidizing the NADPH, therefore reducing your target molecules. In the citric acid cycle, also aka Krebs cycle, aka TCA cycle, you have oxidation of glucose to CO2. So you're basically breaking down glucose. CO2 will be your byproduct. So as you pluck off those carbons, as you go through step by step, CO2 is going to be the byproduct. That's how you get rid of those carbons one by one. In this process, you are making energy. All right, you are basically going from GDP to GTP, and you can further go on from ADP to ATP. You are also reducing NAD plus to NADH, which means you are oxidizing your carbon molecule. So opposite. All right, which of the choices below correctly ranks from least to greatest the contribution of the various ecosystems as to their average net primary production per unit area? All right. So when we look at average net primary production, you're looking at gross primary production minus what you lose through respiration. So basically like what's left over for consumers to eat, all right? And primary production refers to the production of like sugars and molecules and things like that from those producer molecules at the bottom of the food chain. So consumers are going to consume that, and so like what's left over for them to consume. That's the net primary production. So you're thinking about least to greatest, and if it was just least to greatest primary production per year, then the pelagic zone would, the open ocean would actually be the greatest. However, when you break it up into unit area, that open ocean pelagic zone is so vast that it knocks it down. It is not actually the highest. It actually turns it into the lowest. All right, so the open ocean would be the lowest per unit area. You've got savanna, you've got the temperate forest, and then the tropical rainforest. And this also kind of has to do with like diversity of different primary producers of photosynthetic organisms. So thinking about diversity, you have like in the savanna, it's kind of grasses, but not that much diversity. The temperate forest has a little bit better diversity, but tropical rainforest, super, super diverse. Like one of those really diverse terrestrial biomes. So here's a little thing showing you what primary productivity is. You have the gross primary production, like in general, just the amount of energy produced, but then you have energy used in cell respiration, so whatever is left over that you put into the tissues and the fruits and things like that animals can eat and then take for themselves. So we had open ocean, tropical rainforest, savanna, and temperate forest. So the savanna, so this is actually a map looking at net primary production in kilograms of carbon per meter square per year. So the purple means very low, 0, and then the red means very high, 3. So notice that the open ocean is purple. So again, if it was just net primary production per year, the open ocean would be the highest. However, when you break it down per unit area, the pelagic zone, the open ocean, is so vast that it takes it down to like a really low level. And then we've got the savanna area. All right, so here are the savanna areas right here. And notice that, like, for instance, here's savanna right here. Here's the savanna area right here. You've got pretty low levels. Here's savanna area right here. Here's savanna area right here. Here's savanna area right here. And then here's some like over here in this region. All right, so you're still getting purple, maybe a little bit of blue there. All right, so it's ticking up, but it's still really low. Then you get to the temperate forest. So temperate forest is going to be these areas right here. All right, so now you're starting to get into the more green hues for the temperate forests. Sometimes a little bit blue too, but you also start getting some of the green, so it's ticking up even more. And then finally you have, let's see, tropical forest, tropical rainforest. So right here, here, here. Notice that these are the places that have like, you're starting to get into the green and then yellow and a little bit of orange, all right? So here's where you're going to have those tropical rainforests. Here's where you have those yellows and reds, and that means the highest net primary production, all right? In most terrestrial and mammalian species, parental investment is greater for female parents than the male parents. This is because females often court and mate with multiple males. Not really. I mean, there are some organisms that do kind of follow this pattern, but usually the females are not the ones courting, for one. Typically, it's the male that's courting the female. And then usually, like, some of the females will do multiple males just to increase their chances of having babies and things like that. But typically, it's the male that's actually courting. Females choose poor quality mates. Of course not. Why would they do that? Why would they choose poor quality mates? The whole point in the mating rituals, the whole point in like the display of of the males in different species is to kind of be an indication of like good gene quality. So, you know, females will be really choosy because, of course, if they're going to like spend energy to make babies, they want their babies to be good babies. Females are often injured in fighting for mates. Again, usually the males are the ones that are like, they're actually courting the females and then also competing for females and fighting. So that'll be the males. Males are typically polyandrous. This term does not refer to males. So the answer is females are typically K-selected, whereas males are more often R-selected. All right. So parental investment is greater for females than males. So it talked about polyandrous. This is when you have females that mate with more than one male during a breeding season. So it said that males are typically polyandrous, but polyandrous does not refer to how males act. So it really refers to how females act. So it's talking about K-selected versus R-selected. With K-selected organisms, they typically have a low number of offspring and they put a lot of investment, time, and energy into raising the offspring. Whereas the R-selected will have tons of different offspring and don't really put a lot of time and effort and energy into raising the offspring. So this is kind of typical of what you see with females versus males. A lot of times females will mate with the males. The males will leave. They just kind of leave their seed and then go, and the males may try to find other females as well. And so that's like the amount of energy they put into it. And the males are trying to just inseminate as many as they can, whereas the females, once they get their male, then they will basically be growing like a baby, either in their body or, you know, they'll make eggs or something like that. And that is time and energy from the female, so already an investment from the female point of view. And this is just actually an example of sexual dimorphism. So you do see this a lot more in specifically like birds, like a lot in birds. But it is something that you do see in mammals as well. And this is when two sexes have different looks of the same species. So different types of monkeys, you'll see this, or apes, different types of like seals and walruses, lions, like things like that. You'll see kind of a difference in the look of the male versus the female. All right, which of the following is part of the egg-producing structure in plants? This is going to be the megagametophyte of angiosperms. So we're looking for the egg-producing structure. So if you look at, first of all, plasmodesmata, no. Plasmodesmata are like corridors or openings in between plant cells to allow the easy flow of like nutrients and fluids and things like that between plant cells. So no, it has nothing to do with egg producing anything. Then you have the microgametophyte. So when you look at the alternation of generations, the microgametophyte actually gives rise to the sperm, whereas the megagametophyte will give rise to the egg. All right, so it's not microgametophyte. And then you've got the antheridium of mosses and the sori of ferns. So here's the moss. The antheridium actually gives rise to the sperm, all right, whereas the archegonium actually gives rise to the egg, or is part of that structure. So that's not right. Again, just like the microgametophyte, you're kind of leading to sperm. And for the ferns, you have these sori, or each individual one is sorus. It's these little kind of like structures that you see on the underside of the ferns. And this is really where you have the sporangium. All right, so you have spores. Spores will get released from the sori. That will then basically emerge into a gametophyte, and this will be like the different generation, okay, haploid generation. And then from the gametophyte, you can then get the archegonium going into the egg, or antheridium into the sperm. All right, so megagametophyte. All right, a booster dose of a vaccine helps to. It's really whenever you're getting vaccinated, they are looking for antibody levels. They're looking for higher antibody levels that your body responding to the vaccine, whatever was in it, and mounting an adaptive immune response. So you can kind of take it down to A or B, because it's not really doing anything with the MHC recognition sites, and I'll talk about what those are. Maintaining high monocyte count. The monocytes are part of the innate immune system, not the adaptive immune system. And decreasing the number of monocytes. So it's not really, I mean, it does activate them and stuff, but it's not really, I don't think it's really affecting the numbers. It's going to be more about like the macrophages, really. So if you're looking at A versus B, it's talking about blood antibody levels increasing the number of effector cells, or affecting blood antibody levels by decreasing the number of T suppressor cells. T suppressor cells are not really going to be affecting the plasma cells that come from B cells. The T suppressor cells help to suppress, cytotoxic T cells, other types of T cells, and suppress the immune response. But the booster vaccine is not affecting T suppressor cells directly. It's actually stimulating the B cells to become plasma cells and start secreting the antibodies. It also stimulates them to become memory cells. So then the next time you come in contact with whatever that was, the memory cells can start spewing antibody out like super fast, super high rate, and your body can fight it off really easily. So these are the immune responses when you get a primary vaccine and then a booster vaccine. And notice that they're looking at concentration of antibodies. So with the initial vaccine, you get a little bump in antibodies, and then with the booster, it like really goes up. So that's really what you're kind of looking for. You're looking for that increased antibody response. It also mentioned MHC recognition sites by increasing effector cells. So MHC recognition sites are what are found on CD8 and CD4 T cells. So they are T cells, where the B cells are actually what are making the antibodies. So the B cells are really what's increasing. The T cells are not really doing that. Like you may be getting CD4 T cells that are helping to activate the B cells. That's what really will help lead to memory. But like you're not necessarily increasing that specifically. You're specifically looking for an increase in the antibodies from these effector B cells. All right, in mice, one gene regulates color. Homozygous recessive organisms are white, homozygous dominant organisms are black, and some organisms exhibit incomplete dominance, gray fur. A gray male and a gray female produce an offspring of unknown gender. What is the probability that the mouse will not be born with white fur, meaning the probability that will be born with either gray fur or black fur? 3/4. All right, so here is the Punnett square for this. So here's the dad, heterozygous gray fur, mom, heterozygous gray fur. So you've got 1/4 that will be black, you've got half that'll be gray, and then 1/4 that will be white. So you want to know what is the probability of not white is basically 1 minus 1/4, which is 3/4. Also kind of notice the ratios. When you're looking at straight dominance recessive versus incomplete or codominance, the phenotypic ratio is going to be different from the genotypic ratio. The genotypic ratio is going to be 1 to 2 to 1 for homozygous dominant, heterozygous, homozygous recessive. But the phenotypic ratio, typically, if you have only one dominant allele, then you will see that dominant phenotype. So the phenotypic ratio will be 3 to 1. Now, if you have incomplete dominance or codominance, those heterozygotes actually look different from the homozygous dominant organism. So the phenotypic and genotypic ratios are the same, 1 to 2 to 1. The most phytoplankton in a lake would be found in which zone? I had mentioned benthic zones before. Remember, that's the very bottom of the lake, or even the ocean, river, whatever. It's like the very bottom. Aphotic, so phytoplankton are photosynthetic algae. So these guys need light, and so the aphotic zone would not be a zone to find them in, because aphotic means no light, whereas the photic zone would get light penetration. So you're left with limnetic, oligotrophic, and profundal. Oligotrophic is not a zone. It's a type of lake depending on level of nutrients and oxygen. So you got limnetic and profundal. So it is limnetic. So let's see. All right, so here is oligotrophic versus eutrophic. So an oligotrophic lake, cool temperatures, high oxygen, typically low availability of nutrients, especially phosphorus and nitrogen. So basically you have low densities of phytoplankton and then vascular aquatic plants. With the eutrophic lakes, you have warm temperatures, low oxygen availability, high availability of nutrients, especially phosphorus and nitrogen, supporting high densities of phytoplankton and vascular aquatic plants. All right, so eutrophic would be better for phytoplankton than oligotrophic for sure. So now we're looking at profundal versus limnetic zone. Profundal zone is basically the same thing as the aphotic zone. All right, so it's going to be the area in the lake that is not receiving light, or at least receiving very low light. So you'd have reduced or absent photosynthesis, and that's not going to work for those phytoplankton. Phytoplankton are photosynthetic algae, typically floating on the surface of the lake, so they need sunlight. Remember, the benthic zone is along the bottom, and of course this probably doesn't really get much light at all. So it's really the limnetic zone. So the limnetic zone is going to be like that photic zone area towards the surface where you have light penetration, therefore you're going to have a lot more of the phytoplankton. Okay, question 37. Hemoglobin, an iron-containing protein in erythrocytes, binds oxygen molecules. Myoglobin, a protein in muscle cells, is used for oxygen storage. What can be deduced about the relative oxygen affinities of hemoglobin and myoglobin? So you breathe in oxygen in the lungs. The hemoglobin picks it up in your red blood cells, carries it through the blood to the muscles and other parts of the body, and myoglobin is in muscle cells and stores the oxygen. So it would make sense that myoglobin would have a greater affinity for oxygen than hemoglobin, okay? Because hemoglobin needs to be able to give its oxygen up to myoglobin, and then hemoglobin can then actually take up CO2 to then transport it back to the lungs for exhalation. So myoglobin should have a higher binding affinity. Here's a little chart, a little graph showing you percent oxygen saturation and then partial pressure. So notice that even at low partial pressure, you've got a much quicker rate that myoglobin will become saturated with oxygen because it has a higher oxygen affinity. And this is in response to the muscle's oxygen needs. Hemoglobin is the purple line. So this will have a lower oxygen affinity compared to myoglobin. And then you've got fetal hemoglobin. Fetal hemoglobin actually has a higher affinity for oxygen than adult hemoglobin, and this helps the fetus get oxygen from the mother's blood cells. All right, in an experiment, a plant is grown at a particular level of light and in a certain concentration of CO2. So with CO2 constant and light intensity gradually increasing to a present level, oxygen production increases up to a point and then levels off. All right, so CO2 stays constant. As light increases, oxygen production also increases, but then levels off at a certain point. If the light is increased beyond the present level, no further increase in oxygen production is noted. In a second experiment, the same increasing regimen of light is used. So just increasing light gradually, but a higher level of CO2 is supplied. So you get just more CO2 than in the very first kind of scenario. Oxygen production in the second experiment continues to increase beyond the point where it leveled off in the first experiment. From these observations, what might you conclude? So obviously, light is controlling oxygen production, but up to a certain point, all right? And at that point, this is where it kind of leveled off in this first scenario, because the CO2 became the limiting factor. So light was controlling it at first, then CO2 became the limiting factor. And when you increase the CO2, you basically increase that cap of oxygen production, all right? So light is a limiting factor, but not the only one. CO2 is also a limiting factor. CO2 is the only limiting factor? No, light also controls it, so light can also be a limiting factor. Both light and CO2 concentration are limiting factors throughout the time course of the reaction. So it wouldn't really be throughout the time course. It would really be more like light is limiting up to a certain point, and then CO2 becomes the limiting factor. Because the oxygen production is increasing for both of them as the light increases, but then there's like a cutoff point. And then you can either kind of level off depending on if you have low CO2 level, or you can go even higher if you have higher CO2 level. So light is limiting up to a certain point, and then CO2 becomes a limiting factor. And I mean, this just is because of photosynthesis. So obviously in photosynthesis, you've got sunlight, which is the energy source. Carbon dioxide, which is kind of like the food in a way. It's like the source of carbon. And the output from photosynthesis is oxygen, all right? So it needs both light and carbon dioxide. All right, plant systematists have found that monocots were not the first flowering plants to evolve. This would suggest what? Evolution does not always proceed from simple to complex. So there's a lot of misconception that in evolution, you always go from simple to complex. And a lot of times this is true. We went from single-celled organisms to the vast majority of like the multicellular organisms, like all the multicellular organisms you see today. Definitely simple to complex. But there are instances where evolution actually goes from complex and it kind of backtracks to more simple. So here is an evolutionary tree for plants, starting out with like protists and things like that, but you kind of go into plants. Green plants, land plants, vascular plants, seed plants. Within the flowering plants, you've got the dicots and the monocots. So scientists suggest that monocots diverged from dicots. Why is this a big deal? Monocots do seem a little bit less structured, a little more simplistic, I guess. In the stems, the vascular tissue is randomly scattered. The leaves are usually like parallel veins. Embryos only have one kind of like little layering here, whereas the dicots or eudicots, they've got different layers. Two of these guys, the veins can be more net-like. The vascular tissue is arranged more in like more of a ring-like fashion. So these guys seem like a little bit more complex than the monocots. However, the monocots are, scientists believe, diverged from the dicots. So evolution does not always go from simple to complex. An example of this is in flatworms. So flatworms, all right, are parasitic species that evolved from free-living species. So with before we kind of got to like the flatworms that we have most commonly today, we had these early proto flatworms that were free-living. And over time, they kind of evolved to be parasitic. Parasitic is going to be more simplistic than free living. With free living, you've got to be able to find your own food, be able to digest it, to break it down properly, to process it. Whereas parasitic organisms typically are parasites because they can't fully process stuff. They have to have some other organism process it for them or break it down for them before they can actually use it. So they tend to be more simplistic organisms, and so this is an example of evolution going from complex to simple, flatworms evolving from free-living organisms to parasitic organisms. All right, in a certain population of 100 deer, 34 births and 25 deaths were recorded in 1 year. Assuming no immigration or emigration occurred, what is R for that population? This one's pretty straightforward. So you're really looking at net reproduction rate. That's what R is. And when you look at that, you're looking at births versus deaths, and you also can look at you're really looking at reproduction rate. So, what you do to get that is you do births minus deaths divided by the total population size. So in this case, it would be 34 births minus 25 deaths, which would be 9 divided by 100, which is the total population size. That gives you the net population rate of 0.09. So it's going in the positive direction. All right, a patient is suffering from kidney failure. Which of the following symptoms will most likely be present in this patient? This is going to be a higher salt concentration in the blood because filtering salt out and waste out of the blood is a big function of the kidney. I don't really think the blood glucose level would really be affected. That would be more of like pancreatic control, things like that. Uric acid, I'll talk about that. Swelling of the liver, that would be like the liver is the big detoxifier. It's like the filter of the blood. So that would probably be, I don't think kidney failure would really kind of lead to that directly. And then excretion of vital amino acids. If you have kidney failure, you're not really getting rid of things like you should. You have accumulation of wastes. Okay, so with uric acid, this is something that you can get gout from. So a buildup of uric acid, this can actually lead to scarring and damage in the kidneys, eventually causing kidney disease. So uric acid can come from a high purine diet, like if you eat a lot of organ meats, things like that, liver. Can also come from obesity, apparently. So, if you have tissue nucleotide synthesis and metabolism, you can get purine nucleotides and bases that can lead to the formation of uric acid. So 30% will be eliminated in the gut through your small and large intestinal tract, and 70%, 2/3, will be excreted through your renal system, through the kidneys. All right, so when you've got these crystals going through the kidneys, your body can kind of help clean it out, but they can cause scarring and damage to the kidneys. So eventually, over time, you can get kidney disease and kidney failure from that uric acid. So not really necessarily the other way around. So a higher salt concentration in the blood, which would lead to water retention. Water retention is like a big telling sign of kidney issues. Because when you've got your blood getting filtered, all right, in the kidney, these nephrons and the tubules, you have water, being filtered in and out, and you also have salt being filtered in and out. So if your kidney is failing, it is not able to really remove those wastes and salts from the blood effectively. Therefore, if you retain salt and waste in the blood, your water is going to absorb back into the body more often to help regulate that osmolarity, and therefore you'll get fluid retention. All right, an investigator found 150 different diatom species in 1 mile of a stream. None of the species comprised more than 5% of the total population. So you have a ton of different species, and you don't really have one that's like overpowering all the others. In a second observation 25 miles downstream, there were only 20 different diatom species, so much less than the 150, but 2 represented 80% of the total population. So 2 of the species out of the 20 were super dominant. What's the best explanation of these 2 observations? So no, not the stream is bigger. Diatoms are pretty tiny. The nature of the stream bottom changed. Not really. Stream velocity, nah. The main thing is likely organic wastes entered the stream somewhere between the 2 sampling stations. So these are diatoms. These are really cool. Photosynthetic protists. They have glass-like cell walls. They're cell walls made of silicon dioxide, so it's like glass. And they come in all these different shapes and sizes, and it's really cool, a very diverse group of protists. But you have different profiles when you have a healthy stream versus a polluted stream. So a healthy stream, you really do expect to see an even distribution of a bunch of different species, because some species will be more tolerant to the pollution, some will be kind of intermediate, some will be very sensitive. So you expect to see kind of like a good level of sensitive organisms, and then excuse me, intermediate and tolerant. If you have a polluted stream, you are going to see a loss of the sensitive species. They can't deal with the pollution. And you're going to see kind of an overpowering of the tolerant species. They can kind of overgrow, outcompete the intermediate species. So if you get a decrease in diversity and kind of like an overabundance of like only specific species, that's a good indication that you have some pollution going on. Yeah, this is question 19. It was repeated on this test. And of course it was the one that like does not have an answer. So see question 19 for that one. All right, question 44. ATP is an important molecule in metabolism because it is readily obtained from the organism's environment. No, absolutely not. The organism makes ATP. It is extremely stable. No, ATP is very unstable. It will last like seconds, maybe like a couple minutes, but like really not that long. Very unstable. It contains valuable nutrients. No, it is just adenosine triphosphate. It doesn't really contain nutrients or anything like that. Its phosphate bonds are very easily formed, but not easily broken. That is the opposite, actually. The phosphate bonds are not really easily formed. They actually take energy to form. They're very easily broken. So for that reason, they are called high energy phosphate bonds. So this is an ATP molecule right here. It's a nucleotide. So you have an adenine base, you have your ribose sugar, and you have triphosphates. And this is the reason why it's called a high energy bond. These guys are all negatively charged. They do not want to be near each other. They repel each other. And so they can bond, but they are very easily pulled apart, all right? Here is the ATP or energy coupling cycle, all right? So what happens is you have ATP, and it basically gets hydrolyzed with water going through hydrolysis, and you can pluck off those phosphate groups. So you can pluck this one off, you can pluck this one off. And what that does is releases energy that it goes into cellular work. So it can go into like endergonic or energy consuming reactions. And other reactions that are exergonic, that are energy releasing reactions, will actually release energy, and that energy that's released can actually be used to put ADP and the inorganic phosphate back together to make ATP. So it takes energy to build ATP, all right? So it's not easy. It takes energy. But to pull it apart releases energy. So it's not very stable, and these are high energy phosphate bonds. All right, crossing over contributes to the genetic variation of a species by exchanging chromosomal segments between non-sister chromatids of homologous chromosomes. in the production of what type of cell does the crossing over occur? This should be really straightforward. Gametes, of course. Crossing over occurs in meiosis, not mitosis, all right? So a main difference is in mitosis, all of your chromosomes line up single file and your sister chromatids get pulled apart. So you end up with daughter cells that are exact copies of the original parent cell. In meiosis, you actually have two different phases, meiosis 1, meiosis 2. Meiosis 1, you actually have your homologous chromosome, homologous chromatids, all right, because at this point you've gone through the S phase, so you've gone through replication. They actually form tetrads, all right, so they pair up. This is where crossing over takes place, where you have exchange of genetic material between two homologous chromosomes. And then in meiosis 1, they line up in these homologous pairs. Homologous pairs get pulled apart. Then you go through meiosis 2, where now the chromosomes all line up single file, just like mitosis, and the sister chromatids get pulled apart. So only in meiosis does crossing over take place, and only when forming gametes does meiosis take place. All right, when you're forming any other kinds of cells, you are going through mitosis, and then the cells are, probably differentiating or something like that, if they need to be. All right, and then forming sporophytes, that's going to be mitosis as well. All right, in a population of, you have 15 homozygous dominant individuals, 25 heterozygous individuals, and 10 homozygous recessive individuals, the frequency of the A allele is what? This can be figured out actually in two ways, all right? So the first way is you can figure out the frequency of the homozygous recessive individuals, which would be Q squared. So you got 10 individuals out of 50, that's 0.2. So now Q squared, just take the square root to get Q, which is the frequency of the A allele. That would get 0.45. The other way is you can take the number of total A alleles divided by the total number of alleles in the population, and that will give you the frequency of the A alleles. So for A alleles, for the heterozygous group, you have 25 because each of these 25 individuals has one little a and one big A, so you've got 25 little a, plus you've got 10 individuals that have two copies of the little a. So you basically have 20 little a's coming from these 10 individuals because these are all diploid organisms. They have two copies. So that means you have 45 total a's within the population, A alleles, divided by a total number of alleles would be 100. You have 50 individuals that are diploid, so they both have, every single individual has two alleles. So 50 times 2 is 100, and 45 divided by 100 is 0.45. All right, so that is the frequency of the A allele. All right, 47, use the cladogram below to answer the following question. Here's the, excuse me. Ugh, my throat is getting dry. Here's the cladogram we are talking about, cladogram question. So which group represents the monophyletic taxon? All right, that is going to be A, B, C, D, E, F, G. All right, this group right here. And let me talk about why. All right, so this is kind of actually the setup we've got, except the letters are slightly different. A monophyletic group consists of an ancestral species and all of its descendants. So here, this would be the ancestral species. Here's all of the descendants. You also have polyphyletic. This includes distantly related species, but does not include their most recent ancestors. So for that, I would say distantly related maybe A, B, and then D, E, but it doesn't include like their ancestors or anything. So I think B would be polyphyletic. Then you've got paraphyletic. This consists of an ancestral species and some, but not all of its descendants. So notice that it's going up one branch right here. So A would be the ancestral species, but if you're doing all descendants, you would go in this branch as well, but you're only going on this side. So in this case, A and B, I don't think that would actually be any of those because these are closely related, they're not distant, but it does not include the ancestral species. So B, This would be the polyphyletic. DEFG, DEFG, that would be the paraphyletic, because here's the ancestral species, and here's some of the descendants, but then here would be others. HIJK, so HIJK, again, paraphyletic, because H is the ancestral species. You're including kind of this one branch, but not this other one going over here. So A, B, C, D, E, F, G. This is your monophyletic group. Okay, a ribozyme is really kind of how it sounds. It is a ribo or RNA molecule, ribonucleic acid molecule, zyme that has enzyme activity. All right, that's what it is. There are a lot of different examples of ribozymes. For instance, there are ribozymes that will pair or be able to like match to either RNA or DNA molecules, and then when they find their match, they can actually clip the nucleic acid chain. And another example is our ribosomes. Ribosomes or ribozymes, they are made up of mostly tRNA. There are some proteins, but most of it is tRNA, and they have enzymatic activities because they put polypeptide chains. They put amino acids together to form polypeptide chains. All right, when an ant in a colony dies, the live ants will throw the dead ant out of the anthill. If a live ant from the colony, ant X, is sprayed with a chemical characteristic of dead ants, the live ants will repeatedly throw, ant X, not any X, ant X out of the anthill until the chemical on ant X wears off. What is the best behavioral explanation of this ant colony? So if you spray a live ant with some kind of chemical that makes it seem like it's dead to the ants, but it's still alive, they will keep trying to kick it out of the anthill. This is actually an example of fixed action pattern. This is not a learned behavior. If it's negative taxis, it probably means that they would like move away from like the quote unquote dead ant. They're not really learning through trial and error. And then they're not becoming habituated. What's happening is the chemical wears off, like it washes off, they can't detect it anymore, something like that, and then they stop. So this fixed action pattern, this is a sequence of unlearned acts, so like innate acts, directly linked to some kind of simple stimulus. All right, so typically it's unchangeable. Once it is initiated, it is usually carried out to completion. So it won't stop unless the stimulus is gone. Typically, the trigger will be an external cue, which is called a sign stimulus. So in the case of the ants, it was this chemical characteristic of dead ants. This is a famous example, a famous test with these stickleback fish. The males have these red bellies, and they tend to be really aggressive and territorial. And so what this scientist, he was looking at this male sticklebacks and noticed that when a red truck passed by the tank, they would get more aggressive. So he was like, wow, I wonder what's going on. And he realized that they were noticing the red. And that was basically their fixed action pattern of like red means another male, means attack and get aggressive. So he tried a bunch of different stuff. He put these little different models, I guess, with like these little red pieces underneath, and every single time they would get attacked by these stickleback fish. But when he put this model that looks just like a fish but no red belly in there, it was totally ignored. So it was like this red coloring on the underside that was that sign stimulus, that trigger, and that triggered the fixed action pattern of being aggressive and attacking these models or other fish that have these red bellies. Finally, vascular plants share all the following characteristics except development of seeds. So here is a phylogenetic tree or cladogram of plants. You've got the non-vascular plants, and then you've got seedless vascular plants, and then you have seeded plants. All right, so Within vascular plants, seeds is not always found. You have seedless and you have seeded. Other things, alternation of generations. So this is right here. This is what's happening. The generations can alternate between diploid and haploid. So you're getting alternation of generations. You get, spores, you get the gametophyte, then you get gametes, then you get fertilization of the zygote, and then it goes into the sporophyte, which then makes spores through meiosis that are haploid. So it's just alternating between diploid and haploid. Dominant to the sporophyte, that is something that you find in vascular plants. So in these seedless vascular plants, you have sporophyte generation that's dominant, as well as the seeded plants. When you get into the non-vascular plants, it's more of the gametophyte generation that's actually dominant. Lignin and cell walls, definitely, like once you get the plants that start having these stems that are standing up, they're like the land plants and things like that, then you definitely need some of those support structures. And of course, if they're vascular, then they will have some kind of vascular tissue, similar, either xylem and phloem itself or something very similar to xylem and phloem, because these serve as vascular tissue. The phloem is going to transport like the sugars, and then the xylem is going to be transporting the water. So the answer is development of seeds. All right, folks, that is it for test number 2 in your mock session 1. So I hope you guys did well. I hope that this video helped explain some of the questions, and feel free to send emails or ask questions if you need clarification on anything else.